Thank you for this opportunity to introduce our results about the induction of innate immunity and adaptive immunity using two different vaccines developed at the Center for Genetic Engineering and Biotechnology in Havana. The first vaccine that I will introduce to you to immunopotentiate the innate immunity is based on already a 16 vaccine that was registered in the year 2015 by our center that against chronic hepatitis B, that is based in two virus-like particles, hepatitis B surface antigen and hepatitis B core antigen. Both virus-like particles have the capacity to induce innate immunity through the activation of toll-like receptors 3, 7A and toll-like receptor 2. And at the beginning of the epidemic of coronavirus disease, we decided to reprofile this vaccine to use in healthy people at risk and also at early stage of the disease for the induction of innate immunity. So, as I mentioned, one of the components of this vaccine is the hepatitis B nucleocapsid antigen that have more than 20% of RNA related to protein content. It means that the RNA contained in this vaccine have the capacity to interact with toll-like receptors and induce the interferon stimulation genes that is the base of innate immunity potentiation. As everybody now, during the elderly is reduced the immunological capacity to react against different infection IM, and especially during the coronavirus epidemic, for example in China, as you see in the graphic in the right. So the mortality in people older than 80 years was higher than in other age group. So the mortality was increasing with age. These figures were reduced later at the time that we knew more about the disease and the mechanisms of action, and other anti-inflammatory drugs were developed, but these figures are common for different diseases. So the mortality is higher with age due to the limitation of the immunological system to respond against the infection with different pathogens. The innate immunity, it have also that is activated with these two antigens, so the different toll-like receptors have also the capacity to activate adaptive immunity. So at the time that you induce innate immunity, the human body not only will develop a cellular response and activate the response against foreign pathogen, but at the same time it will be prepared to activate adaptive immunity quicker, faster. So this is why this kind of vaccine, that is a kind of platform vaccine for induction of innate immunity, can be also combined with other immunization approach to increase the immunological response. At the beginning, we conducted a clinical trial in volunteers comparing treated group with no treatment in two groups with 24 and 20 volunteers, and in all the cases were suspect contact of a positive patient with coronavirus disease. The schedule, the administration schedule that we were using was nasal spray in 7 days, so 0, 7, and 14 days, and by nasal route, and daily sublingual administration of the same vaccine formulation containing the two virus-like particles. The volunteers included in this trial were older than 60 years. We were following up these volunteers. The first was to show the safety of the vaccine administration. So 70% of subjects have no any adverse event, and 21% were adverse event, but all these adverse events were mild, mainly nasal secretion, sneezing, fever, asthenia, otology, pneumonia, and hypertension. So most of these mild adverse events were considered not related to the vaccine. As you see here in the next slide, by PCR, we were able to follow the toc-like receptors 3, 7, and 8 before, during, at the end of the administration. Here we are showing the different expression of the different toc-like receptors 3, 7, and 8 at time 0 before starting the administration of the product, and 4 days after the administration of the product. At that time, all the volunteers have only one nasal administration on the first day, and 3 daily sublingual administration. As you see in the next slide, we were able to obtain very significant increase in toc-like receptor 3 in a treated patient, darker color here, in comparison with non-treated patient. So in non-treated patient, the increase of the toc-like receptor was significant lower. The same happened with toc-like receptor 7 and with toc-like receptor 8. We were not able to obtain significant increase. So the P was not significant for toc-like receptor 8 in these oropharyngeal scraping samples. So the overall statistical estimation with the 3 toc-like receptors at the same time was very significant comparing treated and non-treated patient volunteers. And at day 8, we obtained very significant increase of 3 toc-like receptors, 3, 7, and 8 in all the volunteers. As you can see here, of course, the overall increase combining the 3 receptors at the same time was also very significant. So it was very clear the capacity of the vaccine in oropharyngeal scraping samples to induce the activation of toc-like receptor as a marker of innate immunity. We also were measuring the activation of lymphocytes and monocytes in peripheral blood, and that we are showing here was a significant increase in the day 4 already of the HLA-DR expression, so that correlate with the activation of monocytes. It's in this way we are preparing the adaptive response to the pathogen in a better way using this nasal immunization with this vaccine candidate. Then we observe the same in lymphocytes in peripheral blood. Was also significant increase of HLA-DR receptor in lymphocyte, showing the activation of these cells. So as a conclusion of this part, the HeberNasvac candidate induce an increase in the gene expression of RNA receptor at the local site of SARS coronavirus 2 infection and the stimulation of expression of HLA class 2 in monocyte and lymphocyte in peripheral circulation, evidencing a superior condition of the immune system of aged patients to respond to coronavirus infection. So at this time we are conducting a large field trial in one municipality of Havana city with healthy people for in older than 60 years, but at this case we are following the infection with any kind of respiratory virus or acute respiratory disease in population. So, and the second part of this talk is related to the vaccine candidate Mambisa, is CIGB 669, that is a specific vaccine against coronavirus that is administered through nasal route. In this case, we are using the same nucleocapsid particle from the HeberNasvac vaccine, so the previous vaccine that we were showing for potentiation of innate immunity, and this nucleocapsid virus-like particle we are combining with the receptor binding domain expressed in yeast, in Pichia pastori yeast. So as was mentioned before, the RNA content, the high content of RNA in this nucleocapsid particle is able to induce innate immune components like toll-like receptor 3, 7 and 8, and we use the capacity of this virus-like particle as immunopotentiator for nasal route. As is known, the nasal route has the advantage to stimulate both the mucosal immunity and the systemic immunity. So the intranasal vaccination has the capacity to stimulate immunological response in both levels that differ from systemic immunization that also induce immunological response at the systemic level. The advantage of the nasal route of immunization, in addition to the systemic response that we already mentioned, is the capacity to induce tissue-resident memory cells, and this is at the same route that the respiratory virus will use for the infection. So it's this way we hope to achieve sterilizing immunity in the subject immunized with this vaccine. There is a recent paper published in Science that include this vaccine candidate as one of the 7 vaccine candidates that were underdeveloped for nasal immunization or mucosal immunization against coronavirus disease. and this was the only one vaccine candidate based on subunit platform. It means a recombinant protein platform. The other vaccine candidates under development are live vector, mainly live vector, and also one attenuated virus. With this, for this vaccine, we were testing 3 different delivery devices, 2 spray devices, and this spray device based in a pump, and also a syringe with the conical device with the capacity to make the spray, and also another device for drops. And for nasal sample, we were using this synthetic adsorbent matrix that is commercialized by Mucosal Diagnostic Limited from UK. This was a phase 1 clinical trial that we conducted in healthy volunteers. We were comparing several different arms, but we show here, this was a randomized double-blind clinical trial, but that we show here is the group that received 3 vaccine administration 2 weeks apart, 0, 14, 28 days, only with the nasal vaccine candidate. And in this group, we were measuring EGA and the EGA in saliva, and that we obtained is that 78% of the volunteers developed EGA antibody titers in saliva. It means seroconverted for at least with 4 times increase for antibody titers in saliva. Then we also showed the capacity of the RBD, the receptor binding domain, expressed in yeast to induce cellular response, interferon gamma secretion in ELISpot with lymphocyte purified from peripheral blood of convalescent people. So the recombinant antigen was able to induce cellular response in lymphocyte from convalescent with at least 3 months of releasing from the hospital. In the next slide that we show is the increase of EEG serum EEG titer 14 days after the administration of only 1 vaccine dose to the convalescent volunteers. And that you see here is significant increase in antibody titers from 6 at time 0 when the vaccine was administered to 206 2 weeks later. In the next slide, we show the inhibition capacity of this antibody. So a functional test where we can follow the increase on the percentage of inhibition, that from 21 at time 0 to 81%. Also significant increase in inhibitory capacity of the receptor binding domain to the binding to the ACE2 receptor. And down you can see the same, but in this case is a titer, inhibition titer, that also increase significantly from 32 to 1526. And in the next slide that we show, it's in this case using the device for nasal sampling, so that that is better than saliva. So in different experiments we confirm that the quality of the sample taken with the nasal device, that is a kind of a porous matrix, is better than the saliva to measure both the IgA and inhibitory capacity of the antibody. And you can see here is the increase in the inhibition capacity of the antibody in the nasal samples. So in this case is an increase in 54% of the individuals, the volunteers included in this trial were able to increase the inhibitory capacity more than 20% in nasal samples. As you can see, there are subjects with very high inhibition capacity in this kind of samples. So at this time we are conducting a phase 2 clinical trial in convalescent individual, and we are going to start. We already submit to our regulatory authority the proposal for a clinical trial in healthy volunteers already immunized with a systemic vaccine. This way we are developing this kind of mucosal vaccine candidate both for a booster of convalescent population and also as a booster of the any kind of prophylactic vaccine, normal vaccine schedule used for healthy volunteers, with the advantage of this vaccine candidate to induce mucosal response and tissue resident memory cell. That is the goal of this kind of vaccine that have shown the safety and the advantage of the friendly administration of the vaccine. Finally, I would like to thank all my collaborators that have been working both at the hospital Luis Diaz Soto in Havana, that is the main hospital in Havana, attending patients of coronavirus disease, and also all the collaborators from the Center for Genetic Engineering and Biotechnology involved in different experiments that I was showing during the presentation. So thank you very much for your time and for the opportunity to share our results. Thank you.